You want to put your phone?
Sure.
Can I hold it here? You want to pop up for that?
Oh yeah, thank you.
I'm okay with that.
Django recording.
I'm just telling you, that's just to have another helper. You can have all the extras. 'Cause I'm
What's
not-
ours?
This one? Okay, let's take a look at what happened with the UV first. Okay, so that's pretty.
You did the pre.
No. Okay, so that okay. Let's look. So for the prion group, this was the long one, right? So here is the control, and you can see that the organism grew very well on here. Okay, good.
Yeah.
Here is 30 seconds, and you can see it killed everything. So it worked really well. Oh wait, there's one here.
Oh, okay.
And then this is at a minute.
This is UV?
This is UV. This is that long one
Oh, yeah,
that I
yeah,
brought
yeah.
in.
Oh, this
So
one we used.
The huge one?
265.
265, that's correct. What's my objective?
UVC.
So the UVC, that's right. So be sure you know UVC, be sure you know 265 nanometers, be sure you know thymine dimerization, be sure you know endospores can survive, be sure you know that the shadowing will protect it, right? So that UV has to be able to reach the cells in order to destroy them. Okay? So the long wand with the prions, very, very well done. Let's look at, this is the bacteria. So the bacteria, they did, what did you all have?
We used the other one.
I'll use this one over here.
Yeah.
Okay.
You didn't, the little one didn't work.
Yeah.
So that one is really interesting because it killed all the cells. There's one here at 30 seconds, but then there's some exogenous cells that happened. So for the most part, it killed everything. These things in here, Victoria, these little colonies, there's something in the air. So it's not anything that we were testing with. So that's after exposure. So we can say that UV light worked really well also. The morning one, I don't know what happened to that little as seen on TV thing, but the morning did that also, and it worked really well. And they were supposed to keep them, but I think they threw them all out. Another over there. You want to go grab those for me, Cheska, somebody? So just the ones that are from the tan colored ones. Perfect. Thank you. So here,
Sorry.
You said 5 minutes.
so this is the as seen on TV. Right, here's a control. Perfect. And then here, those little things work. Here you can see they knocked everything out. There's a few at 1 minute that survived. And then there's a couple over here at 30 seconds, but they were on the side. And then there was some exogenous stuff. She likes those big words. Okay. So you will be tested over not what happened with our little experiment, but you'll be tested over the wavelength, the type of UV, the mechanism of action, and then endospores and shadowing and
Basically the
I
list
might-
you just ran through.
yeah. But I might remember we went over this and I showed you like 1 minute, 3 minute, and I might ask some questions about that also. Okay, good. All right, now. This one was the virus. No, is this virus group?
Yeah, it's ours.
And you guys used the.
The one that you plugged into the
UV.
computer.
USB thing.
It didn't work.
Oh.
and this is the second time it didn't work.
Oh, boo.
So it correlates with the data we saw in the morning that it just doesn't work, right? So
Like, why? Like, it just doesn't work
because they
anymore?
sold me something that didn't work.
Oh, okay.
So this,
that's
so you know, I'm glad I know this because I'll probably get rid of those things because they don't work clearly.
Which
ones?
So it's not our
This is a
fault.
little, no, that was a little, the little short one,
The one you would take home?
Oh, the one that, the
Just plug
USB
in.
port.
the
Yeah.
USB one.
So you think that was just that particular one or all of them?
Well, I have another one, so I bought
Okay.
two because they were like 2 for 1. I was wondering why they were 2 for 1. But so it's clear that they don't work. Clear.
Yeah, so
Right.
all those colonies then.
So
Huh.
sometimes you buy things and you find out they don't work. Sometimes you don't have the ability to find out they don't
Right.
work. And
Most-
you're using them
most
the whole
people-
time and thinking they're working.
How
You
much
didn't
were
buy it
they?
from Shop LC, did you?
What?
You didn't buy it from Shop LC, did you?
No.
Oh, okay, because that's where I work.
Yeah.
Nobody's stuff.
No, no.
I'm dead.
So I'm gonna let you all look at those, okay? So let's look at the antibiotics. And so if we look at the Piron group, the Piron group had really good results. You can see, look at how beautiful
Oh,
those
wow.
were.
Nice.
Yeah, nice.
So let's look at these for a minute.
Mhm.
So if we were to look at those, you could see that this one, this one, this one, this one, were probably something we need to measure
Mhm.
to see,
Yes.
this is E. coli,
Right.
to see if they would be effective against E. coli, right? What is the area around the
Zone of
disc called?
inhibition.
Zone of inhibition. What is the growth on it called?
Lawn?
A lawn, or confluent growth,
Oh, I
right?
didn't know that.
A lawn, yeah, a lawn.
LAWN?
Uh-huh, like you mow
Like
it.
a lawn.
Oh.
Uh-huh.
Oh, okay.
Okay?
Or
Oh, that's
confluent
cute.
growth.
Okay.
So, what is the standard that we use to make sure we have the right population?
What's the standard that what?
That we use to make sure we have the correct population?
You mean the correct organism? What do you mean population?
So remember I showed you a standard, the turbidity standard?
Okay.
What was it called? Geo, middle, open up the middle cabinet on the left, good. And you see some little test tubes right in front of you?
Oh,
Pull one of them
McCartney?
out, pull one of them out.
Yeah, that one.
Yeah, that one's good.
McFarlane?
That's a McFarlane, that's right, McFarlane.
That's what I was trying to say.
So, this is a McFarlane standard.
When I said I was.
Remember that we said that the amount of turbidity that is in the population is directly related to how many cells. You sure you know what a McFarlane standard is and why it's used? Okay? What is the process called of streaking and doing the susceptibility testing? Who are the famous scientists that came up with it?
Mansfield?
Nope, that was the
That's
person
the
who came up.
groups. Oh, Kirby and Bauer?
Kirby Bauer, right, Kirby Bauer, right. And so we use Kirby Bauer so you can see somebody did this and somebody did this. This one was a Staph aureus, and boy, just about every one of these things worked, right? Now we'd have to measure the zone in order to figure out if they're effective or not, right? What would you do, Miss Carolina, if you didn't appreciate the end of the zone on this side? What would you do to figure out what the true diameter was? Come closer so you can see it. So with this one here, right, you can see that you've got from here to here, but you can't really tell where it goes here. What could you do?
Well, I should have put them further apart.
No, it's okay. But what you would do is measure from here to here and then multiply it by 2,
Yeah.
right? And so then you could find out the total diameter. So these are problems we have in the lab, right? Not every time is everything going to be perfect, and so what we need to be able to do is to use our foundation in science to figure out a problem, right?
Our note said don't use more than 4 discs per slant though, so what's up with that?
No, I said up to 6, but I said just target 4.
Oh,
That was not my miscommunication. It
Okay.
wasn't. So I did 6.
Yeah, we did.
yeah,
six.
we did a lot.
That's okay. That's okay. There's nothing wrong with this. I mean, these are actually well done. And so you can see here from the ones that were done. Let's see, what is this?
Oh,
Why, Cody? Why?
right?
Staph aureus. So you can see that on Staph aureus, clove really worked well. Right? That's not bad. And then for E. coli,
Okay.
clove, and tea
Let's
tree.
raise our hand.
Uh-huh.
Right?
Clove and what?
Tea tree.
Tea
Tea
tree.
tree.
So the only other one that would have been different would have been what the Chescanator did. And so let's look at hers. Oh, that's nice too.
Oh,
You see?
nice.
Yeah, it's really nice. Here's one that shows that Lysol was really, and bleach were pretty good. And then here's what the Chescanator did. So,
What happened to those dogs?
well, so the ones that worked were the C. What was the C? Do you remember?
Clove.
Clove. Clove is all over the place. And the other one is DF,
Douglas fir.
Douglas fir. And F,
Frankincense.
frankincense, which I liked. And S had a little bit.
That's a C. Grant.
S.
What was S?
We don't know. You tell
I
us.
have the picture. It's on my phone.
Okay, okay. So you can see the way they worked. This was against Staph aureus. So there is a little bit of something other going on here, where I think this is the one we did first, where there was a little bit of
A lot of oil.
some of it kind of.
Yeah. This
But
yeah.
that
The other
that
ones.
area, it's not showing up really well, but maybe I can find one real quick. Hold on a minute.
With the antibiotics test,
Yeah.
how can we tell which antibiotics they are? They just look at the little disc.
Yeah, each one of them has a little, like a little.
Can we look it up online and it'll tell us?
No, you would have to look at the
Individual
bottle.
sticks?
I mean, it might tell you. Let's see. No, none of these have breakthrough. So breakthrough, I don't have a good example of it, but can you see right here that there's colonies growing in that little area of clearing? That's called breakthrough. And in bacteriology, when you have breakthrough, you should never use that antibiotic.
Why?
Because there's some organisms in that particular culture that are becoming resistant to the antibiotic.
That's what it means. That there's.
Well, that's what it means bacteriology. When they say there's breakthrough cases of COVID, that means that the vaccine isn't protecting enough that the people got infected. And that's normal, right? But people get all excited about it. But that's what they mean by it. Yes, ma'am.
Cheska needs clarification on susceptibility and resistance.
On susceptibility and resistance? So susceptibility would mean that there is a zone of inhibition, and the zone of inhibition shows that the organism is sensitive to it.
Okay.
Resistance means that the organism grew right up to the disc,
Right.
and that particular antimicrobial agent had no effect on the cell.
Okay.
Okay?
Uh-huh.
You never want to use an antibiotic that the organism is resistant to, because it
It
won't
won't
work.
work. Okay.
Okay? So there will be no write-up for this. You are welcome to take these and look at them. This is the bacteria group. This is the virus group. Oh, and I mixed yours up. That's the prion group along with.
Yeah.
Now I'm all confused. Carolina, can you help me? Which one are yours? This one's yours, and then which one of these?
Yeah.
The blue ones are
Everyone's handwriting.
For real.
I'm sorry, I mixed them up pretty wrong.
This one's mine.
Exactly what
Okay,
I
pull it out
did.
then.
I was like, fuck.
These are for sure okay.
I got some.
I don't know the rest.
So I guess these are,
Do you guys want to go pull them out? Which ones were
that
y'all's
yours?
handwriting? I don't know who was here that day. You.
so
Yeah.
these are you all's, and then these belong to you all.
Are these y'all's handwriting?
Very interested in what mine looks
So
like.
take about 5 minutes, and then
didn't
we'll
do
review
that.
for the practical.
We did that?
Yeah, CT right here.
Okay, whose is this?
Cloud, yeah.
That's probably
Because I did too, didn't I? Go.
I
This
did-
one is,
I did
'cause I
too
wrote
as
it.
well, but I remember doing
probably from the.
I think I did two.
Right, we did. No, I did one, right? Because he told me.
Okay, I did one too, because there was one I didn't pay attention to, but I didn't
Okay,
think
so
it was
this is.
100%
Oh shit.